Okay, so hi everyone. My name is Rachel and I'm the head of adult services at the Brookfield Library. And I want to welcome you all to our program, What are Cyanobacteria and Why Should We Care? Our presenter this evening is Dr. Ed Wong, who is Associate Professor of Biology at Western Connecticut State University, or WestCon. And he's been managing a program over the last 6 years to monitor and investigate cyanobacteria in Connecticut recreational waters. So without further ado, I will turn it over to Dr. Wong.
Hi, everybody. I'm going to first make sure I share my window. Whoops. So let me begin by telling you a couple of stories. Stories are always good to use. So it was a warm August day in North Carolina when Melissa and Denise took their 3 dogs to a pond to play. When they got home, one dog started seizing and foaming at the mouth. The rest soon followed with the same symptoms. Though the dogs were brought to the veterinary ICU, none of them improved and all 3 were dead within 24 hours. The veterinarian told them that blue-green algae had sickened the dogs, causing them to have internal bleeding from livers that shut down. Francis Connors and his 6-month-old German Shepherd, Kiana, were playing in the Virgin River on Zion National Park when Kiana began seizing. She died later that day. The health department detected high levels of anatoxin produced by toxic cyanobacteria in the river. Dane Rogers was looking forward to his senior year in high school, where he was captain of the soccer team and a member of the National Honor Society. But on this hot July day, all he wanted to do was swim in the pond at the local golf course. 2 days after swallowing scummy water, he died. Samples from his body showed the Cyanobacterium anabina and its toxin, Anotoxin A. Dane's father said, "Never in a million years did we think something like this could happen." So welcome to my talk about cyanobacteria, also known as blue-green algae in some places. And I want to talk a little bit about what cyanobacteria are and what they can do or what they have done, and also what can we do or how can we respond and what can we do about it. So a couple of more news articles, headlines for you to get you nice and juiced up or panicking. Blue-green algae outbreak kills 32 cattle in Oregon. Blue-green algae blamed for deaths of thousands of fish at Lake El Reno in Oklahoma. Huge toxic algae bloom sickens more than 100 in Utah amid a heat wave. Here in Lake Erie, especially in the city of Toledo, they've been hard hit for several decades now with toxic algae blooms or cyanobacteria. And you can actually see in the satellite picture the bright green cyanobacteria that's up against the western shore of Lake Erie. And in fact, in 2014, the city had to shut down its public water supply for 500,000 people because of cyanotoxin contamination. And then back in 2019, Mississippi actually had to close down its Gulf Coast beaches because fresh water coming out of the rivers that were contaminated with cyanobacteria were spilling out into the Gulf. And of course, who could forget New Jersey's largest lake, Lake Hopatcong, shutting down in 2019. Probably, I don't know if it's the first time it's happened in recent history, but first time I've ever heard of it happening, and it was because of cyanobacteria blooms that were appearing throughout the lake. One of the most tragic stories I've ever read, and this was in the scientific literature, was about a kidney dialysis unit in Brazil that inadvertently used a water source that was contaminated with microcystin, which is one of the cyanotoxins. And needless to say, flushing renal patients with toxin-laden water was not a good deal. As you can notice from the headlines there, 100 suffered liver failure and 52 people actually died from it. So it was probably one of the biggest cases of cyanobacteria poisoning of humans and causing deaths that's been recorded. Now, besides just immediate, acute poisoning, one of the big concerns that's cropped up in a number of investigations is that long-term low-level exposure to some of these toxic cyanobacteria could be doing things that cause some of these neurodegenerative disorders, such as amyotrophic lateral sclerosis, also known as Lou Gehrig's disease, or other types of things like Alzheimer's disease and so on. One study from New Hampshire noted that people who lived around areas, around bodies of water that suffered high levels of toxic cyanobacteria had much higher rates, much greater rates of these types of neurodegenerative diseases later on in life. And as you can see in that lower headline there were dolphins that were found that had these amyloid plaques in their brains, and also the associated cyanobacterial neurotoxins in their bodies. And amyloid plaques, as you know, are one of the classic signs of Alzheimer's disease. Just recently, this past year, after a 25-year hunt, some scientists finally determined the cause of a large number of eagle deaths that had occurred. And they ultimately determined that, they found the smoking gun actually, they found that it was a cyanobacterial neurotoxin that was causing the brains of these eagles to basically just kind of wither away. They were able to show that a certain species of cyanobacteria produced the toxin. The toxin was shown to specifically destroy parts of the brain, destroy the myelin sheath, which is part of the insulation of the nervous system. And so they were able to sort of definitively confirm that all these eagle deaths over the past several decades were due to this cyanobacterial neurotoxin. Quite an interesting biological detective story. So let's get sort of the basics. What are cyanobacteria, also known as blue-green algae? And I hate the term blue-green algae, because even though it's been around for a long time, because algae traditionally refers to plants. Algae scientifically are plants, but cyanobacteria are bacteria. They're not algae, right? The bacteria, as you know, are a different type of form of life. They're much more simple form of life than plants are. The second thing is that cyanobacteria are ubiquitous throughout nature. I mean, they're just found in all terrestrial environments. They're found in water, they're found in soil. We cannot get rid of them. They're part of the natural ecosystems. And they make oxygen through photosynthesis. That's why they're blue-green in color, and that's why they're called cyanobacteria, because they're actually photosynthetic. So in fact, it's been believed, they've actually found fossilized mats of cyanobacteria that date back, literally to the beginning of life, the appearance of life on this planet. And it's believed that, in fact, the cyanobacteria were the ones who actually made the first oxygen atmosphere for our planet, and continue to contribute a certain amount of oxygen to our atmosphere right now. Cyanobacteria, some of them at least, are able to fix nitrogen. And what that means is that they're able to take nitrogen gas out of our atmosphere and convert it into useful nitrogen compounds such as ammonia and nitrates, which are actually very vital for the production of proteins in all sorts of living things. So nitrogen fixation is a very powerful ability that they have. And also it means that they can make a lot of their own food, right? Besides making sugar from photosynthesis, they can also make these crucial nitrogen compounds that also feed them. Now, you might have come across cyanobacteria in spirulina. If you go to any of these health food stores, you might come across spirulina or some of these other blue-green algae foods that I've shown here. And there's been a lot of concern about some of these things, because as health food nutrients, they're not regulated by the Food and Drug Administration. And of course, one of the concerns is that since they are a type of cyanobacteria, could they be toxic? Could toxic strains of cyanobacteria get into these particular products? And there was one study that was done back in 2020, last year, where they tested a number of these cyanobacteria dietary supplements, and they found that some of these products had more than 40 times the maximum acceptable concentration of cyanotoxins. So the fact that somebody may be growing some of this in a pond in a controlled environment or whatever it is, still you could have toxic species or strains that blow in from elsewhere and start to take over part of that pond, and now they're generating these health food products from it, and that could be very detrimental to the consumer. So if we look at the lifestyle of cyanobacteria, they obviously love to grow in the heat, in the summer. So you'll see that in the summertime, that's when they're mostly growing. And as the weather starts to cool through the autumn and into the winter, many of them will form little cysts, or they'll form little hard shells around themselves. They may settle into the soil or into the sediment, and that's where they hibernate through the winter. And then as the weather starts to warm back up again in the spring, those little cysts or spores will start to rupture, and they'll start to divide again and become, reproducing back in whatever environment they're found in. So the idea that we can get rid of these guys forever, no, we can't, because they're a native part of all these different terrestrial environments, and you'll always have them around. You'll always have some of these cells around. Now, these are some of the major cyanotoxins, and I just wanted to show you the structures of these because they're rather large, and some of them are rather large and complex. The ones that are highlighted in red are the ones that I study in my lab. Microcystin is the primary cyanotoxin that my lab actually measures and tries to measure during the summer. And you'll notice that microcystin is a hepatotoxin, which means that it destroys liver cells. It actually inhibits enzymes that are crucial for liver cell survival, so it can actually destroy liver cells. In fact, for those 3 dogs that I talked about in North Carolina, they died when their livers shut down. They probably were poisoned by microcystin. Now, one of the other toxins that you heard about was also anatoxin, and that you see the structure. That's one of the neurotoxins, and along with saxitoxin. Saxitoxin you might have also heard of before, but may not have registered. It's also called paralytic shellfish toxin. And it turns out that some saltwater microbes are able to produce saxitoxin as well. And that sometimes gets into seafood and paralytic shellfish toxin and causes health problems. So these are all neurotoxins that are actually rather potent. They're very, very potent. As you can tell from the stories where the dogs were just exposed, briefly for a short time, and they suffered, acute organ failure and seizures, and they died pretty soon after that. Now, I became interested in cyanobacteria actually back in 2013, as you can see from this article from the News Times, the Danbury News Times. I read this article in 2013. I thought, wow, I didn't even know that there were cyanobacteria problems in Candlewood Lake. This was, in fact, was a giant bloom that took place in October of all times. So, when the weather was cooling down and the water was cooling down, they had this giant bloom of cyanobacteria occurring in Candlewood. And that kind of intrigued me. Prior to that, I was doing research on zebra mussels. I don't know if any of you have heard about the zebra mussel problem here in this area as well. I mean, they're another invasive shellfish that has taken over the Housatonic River, is now threatening to take over Candlewood Lake, or it's now starting to appear in parts of Candlewood Lake, which really worry everybody because they can devastate an ecosystem. And so I was studying and investigating zebra mussels at that time. And then this thing about cyanobacteria popped up, and I thought, wow, is anybody looking into this? Is anybody studying these guys? Are we measuring the toxin levels? Is anybody studying the genes and the populations here? And so I got in touch with the folks at the Candlewood Lake Authority. And they said, yeah, come on down. We'd love to have somebody be interested in this and maybe do some investigations locally. So that kind of launched me into this thing about cyanobacteria. And then I started a little summer program to test waters and measure the concentration of the microcystin cyanotoxin. And then some of the towns and their health departments said, yeah, we would appreciate having a program where throughout the busy recreational months of July and August, we have the waters tested at our beaches for cyanobacterial toxins. And, would you be willing to do that? And so I set up a program and got support from all these towns and their health departments. And then soon other towns and other lakes started joining in this as well. And so this summer, these are the lakes that I am testing at. You'll see that besides Candlewood and Lake Zor, which were the two first lakes that were involved in my program, Bisig Lake out in Middlefield joined in soon after that, and then Bantam Lake. And then this year, Lake Lillanona and Ball Pond joined in as well. So I've got quite a number of lakes that are involved, and we're each week we test, we sample the water from the beach areas, and we test them for cyanotoxins, and then we report these to the health departments and health department directors and the lake associations, so that they can make informed decisions about what they should do with the beach. For Candlewood, of course, we've got the 5 towns surrounding Candlewood Lake, and they've got their public beaches. Here they are marked on the map. And so we go out and sample. There I am at, that looks like, actually that looks like Eichler's Cove out in Lake Zor. And on Lake Zor, which of course is the impoundment of the Housatonic River, I sample at Jackson Cove, which is the Oxford public beach, and then Eichler's Cove, which is Newtown's public beach. And I recently just applied for a grant and got a little bit of money so that I could also test Kettletown State Park, which actually, if you've ever been out to Kettletown State Park, they've actually closed that public beach for the last 3 years. In fact, they still haven't reopened it ever since we had a really, really bad cyanobacterial toxic season a few years ago. And I'll talk more about that later. But after that, they shut down the beach permanently, and they still haven't reopened it. And it's pretty bad out there, I guess. Well, I don't know. The water's not too bad this season, but it just looks really bad. So here's the season that was pretty bad, 2017. Kettletown State Park. You can see these giant mats of green floating material in the upper right here, and then down in the lower right, you see these kind of streamy green things in there, right? Those are just billions and billions of cyanobacterial cells floating around in the water, and that's not a good sign. Here's a picture of Eichler's Cove, and this was really bad. I mean, look at this. It's like pea soup green. It's almost, I often say that it's like somebody just dumped a can of green paint into the water, and that's what it looks like. So in situations like this, usually the town health department will shut down the beach, and they'll post signs like this that say, the beach is closed due to high bacterial levels. And it's not just for fecal bacteria. This has nothing to do with fecal bacteria. This actually has to do with cyanobacteria and the toxins that they could be producing. I'll talk more later about some of the concentrations we've detected there. So Bisig Lake, Middlefields Town Beach, I go out there and sample. Here's a picture here on the left of my boot and a big patch of cyanobacteria floating in the water. Actually, I took this picture back 3 years ago. And frankly, I just took a picture just like this last week at Bisig Lake. So it looks pretty bad too. It looks just like that, in fact. Now one of the things you look for is this characteristic line of cyanobacteria washed up on the sand right at the water level, right at the water line. And here's a tube over here on the right where I scooped up some of the cyanobacteria. And you can see that it's just really thick with lots and lots of cyanobacterial cells. So how exactly do we monitor these cyanobacteria? Well, there's 3 things that we primarily do. We do microscopic identification, so we can try to figure out, what kinds of species or genera are present. We screen for their genes. Now remember, genes are the genetic information that determines the production of pretty much anything in all of us. But we're looking specifically for the genes that are involved in producing cyanotoxins. So if we can find the genes in a population, it means that that population certainly has the genetic capability of producing these toxins. And then finally, we measure the toxins directly, right? We measure the cyanotoxin concentrations, and that helps inform us and also the health departments about whether or not the conditions are dangerous for recreational users. So real quick, photomicroscopy. Some of my students and myself have been snapping pictures of these for the last couple of years. And actually cyanobacteria are really pretty. I mean, if you look at these pictures, you know, microcystis is kind of blah, it's just a big clump of cells, but some of these others like Delicospermum, Anabina, Oscillatoria, Planktothrix, Warrenicinia, they're really cool looking, you know, and especially I think the Delicospermum, the spirally Delicospermum are really beautiful. And, you know, and they're just all different kinds of different shapes and sizes, and they're really neat to look at. So in terms of detecting toxin genes, we use a procedure called PCR, which you may have heard about. I think maybe more people have heard about it now because PCR is the method that they use to screen people for the presence of the COVID virus, right? Except we use PCR to screen for the presence of the toxin genes that are present in these cyanobacteria. So the way we do this, we collect a water sample that could have a whole bunch of different types of cyanobacteria in there. We isolate all their DNA from it, from all these cells, and then we use PCR to specifically amplify the presence of any toxin genes that are there. So that's what PCR does. It only makes lots and lots of copies of something that is there, but if there are no toxin genes present in the genomic DNA, then of course PCR will generate nothing. It'll say there's nothing here to make copies of, so you have no toxin genes present. And then we look for the presence. If the toxin gene is present, we'll see the products of PCR. If no toxin genes are present, we won't see anything. And so that's how we screen for these toxin genes. So just a real quick thing from last summer. I mean, I've got years and years of data, but I just want to show you something real quick and make it really simple. So wherever you see an M present, it means that we found the gene for microcystin. That is the gene that produces the microcystin toxin. And you can see this throughout the summer of 2020, throughout July and August. Pretty much microcystin is found, those genes are found in all the cyanobacteria populations. Excuse the one basic sample here that has kind of empty. We got to repeat that sample, something went wrong there, but it's a pretty common gene that's found in populations of cyanobacteria, which means that this toxin can be produced in pretty much all of these places. Now saxitoxin, that one of those neurotoxin that's present, it's a little bit more rare, but you'll see that it's found in certain places. Wherever it's white, that means that we didn't find that gene present, and ND means it wasn't done. We haven't tested those samples yet, but you'll see that Brookfield, Danbury, and Eichler's Cove seem to be kind of hotspots for the presence of saxitoxin, for the saxitoxin gene. Now, let me just say real quickly that, and I'll repeat this later, but we don't actually test for the presence of this toxin. In fact, there aren't that many places that test for this toxin. Certain states do, but this is not tested for here in Connecticut because it's believed that it's kind of rare, but in the absence of testing, we don't really know how much saxitoxin is present in these particular waterways. Anatoxin is even more rare in terms of the gene being present in these populations. And you'll see that most places do not have the anatoxin gene, but Brookfield, yay, you win the prize because you guys seem to have a lot of anatoxin, at least for the days that we sampled, the weeks we sampled. So Brookfield, for you folks who live in Brookfield and use the Cadogan Park Beach, might want to be a little bit more careful. Not sure exactly. Again, we don't test for anatoxin in the state of Connecticut, not normally. And so we don't really know what the, how much antitoxin is present there. Besides the presence of it, we also test how many copies of the gene are present. So what that means is if you have a lot of copies of the genes, it means that there are lots of cells that have that gene and they may be producing the toxin. So this is kind of a heat map picture. You can just kind of ignore some of the fine details, but you can see that the warmer the color, the higher the number of genes that were present. For example, in Eichler's Cove, out here on the 12th of August last year, there were a lot of cells there that had the microcystin gene. Right? But if you look at Brookfield seemed to peak out so in July in the 900s, and then it kind of petered out a little bit, and so on. So it's quite variable in terms of the number of gene copies that are present. Saxitoxin, Brookfield. Yay, Brookfield, you're back in the news again. So Brookfield had very high numbers of saxitoxin genes present in July, and then it kind of petered out. In fact, almost everybody kind of petered out throughout the rest of the summer. So it looks like this gene, or rather the cells that carry this gene, seem to be very prevalent early in the summer, but they kind of, disappear or go somewhere else later in the summer. They just start to die off. Not quite clear what's going on there. Antitoxin, well, again, antitoxin doesn't show up very often. It does show up in Brookfield and Danbury. Bantam, which is the bottom two rows, right? They seem to have a lot of antitoxin. Remember, antitoxin is the neurotoxin that killed the dog in Utah, and probably also the high school boy who was swimming in that golf course. Now, this is an important figure. I don't want to try not to confuse you guys too much, but what I'm trying to point out here in this picture is that not all cells, not all cyanobacteria carry toxin genes. And I think that's an important distinction that we have to understand. Just because you see, the water all slick and green and thick, doesn't mean that those guys actually carry the genetic capability of producing toxins. Similarly, the other flip side is that even if you don't see a lot of cyanobacterial cells, they could be carrying toxin genes, and they could be cranking out lots and lots of toxin. So in this picture, you'll see the blue bars represent what percentage of the cells in that population have the microcystin toxin gene, right? So you can see that Jackson Cove, JC right here, had like almost 90%, 85% of the cells in that population carry the toxin gene. But then if you look on the light green bar, which is the amount of toxin produced, you'll see that the toxin levels are actually rather modest. Whereas in Eichler's Cove, which is not too far away from it on the Housatonic River, right? 50% of the cells had the gene, but they produced a lot of toxin. So if we look at Brookfield, Brookfield has a rather high number of cells. 40% of the cells from the Brookfield sample have the toxin gene, but the amount of toxin that's produced is rather low, right? So what this tells us is that even if you see cyanobacteria, it doesn't mean that they're toxic. It also means that even if they carry the gene, they may not be using that gene. That is, they may not be actually turning on that gene to make toxin. So this makes it really tricky. People have always kind of assumed that, if I see cyanobacteria, they must be producing toxin. Or if I detect toxin genes in them, they must be using those genes and making toxin. And what we found, and also other researchers have found, is that no, there's certain triggers in the environment that turn on these genes to make the toxin. And we're still trying to figure out, all the different parameters and triggers and the environmental triggers that might be involved in that. And so it makes it really, hard to just visually look at water at your beach and say, oh, this is dangerous. Well, we don't know if it's dangerous until we actually test the water for the toxins itself. Okay, so hopefully hopefully that I made that clear. So how do we measure toxins? We use what's called an ELISA assay, which some of you may also have heard because they use ELISA assays to test for COVID virus, right? That's another thing that some of these techniques I use might be familiar with people, because we keep hearing these terms when they're talking about testing for COVID. And we use the same kind of techniques, except we test for something completely different. So I want to point out what the US EPA, the Environmental Protection Agency, has set up as the threshold for danger, essentially, the threshold for allowing people to use recreational water. So you can see that in 2016, the EPA decided that 4 micrograms of toxin, of microcystin toxin per liter of water, which is also known as 4 parts per billion, is the limit that they will set. And if you exceed that limit, then you've got to tell people to get out of the water and stop using that water. In 2019, with a different administration and also change in the EPA, they raised the threshold up to 8 micrograms per liter, or 8 parts per billion. So in other words, they made it, you can get away with more toxin in the water before you have to tell people to get out. Now, let me just say a little bit about why there was that change, and they'll say, well, it was due to our investigations, but there's a lot of talk that believe it or not, the cattle owners associations in the West were a major group that kind of lobbied for a higher limit. You might wonder, well, what do cattle have to do with cyanobacteria? Well, cattle poop is a nutrient that if it gets into the water, it feeds the growth of cyanobacteria. And of course, cattlemen and cattle people, cattle owners, they'd rather not have these very stringent rules that prevent them from having their cattle poop all over the place or contaminate waterways and so on. And so they actually wanted, right, a higher limit so they can continue to let their cattle dump their waste into waterways. And so there was some of that at work as well. But anyway, it's not quite clear which of these is really is 8 micrograms per liter safe? Is it low enough that it's not going to be a big deal? And there's there's arguments on both sides about whether that's proper or not. Again, we don't fully understand the consequences of low level exposure. It's just like smoking, right? You can smoke for 20 years and all of a sudden cancer appears. And we've already seen some of these suggestive studies that indicate that long-term low-level exposure to some of these cyanotoxins will lead to brain neurodegenerative diseases and things like that. So there may not, we don't really understand the full safe limit for these things, and that's true for lots and lots of toxins, in fact, in our environment. So you'll see that in when I talk about our results when we tested water, that sometimes, prior to 2019, we'd say, oh, it was over 4 micrograms per liter, beaches have to be closed down. But then after 2019, with the higher threshold, we had to say, well, it's over 4, but we can't legally tell you to close down the beach. Although, good health directors will say, it looks bad, it looks very green, we should close it down just from a visual perspective. So I throw up this chart to make your brain bleed, right? To make your eyes bleed. No, here's the take-home lesson from the summer of 2016 when we first did our tests. All samples were less than 1 part per billion, right? And this is back when the limit is 4 parts per billion. So everybody's okay throughout the whole summer. Nobody exceeded the threshold, although we definitely could detect levels of cyanotoxin in there, right? So if we look at Brookfield, well, let's see, I think July 14th, it was the highest level it got up to, which was 0.733 parts per billion. Okay, still less than 1 part per billion. Everybody's okay, at least according to the EPA, we're okay. We don't have to close the beaches. Get to the summer of 2017, and all of a sudden, Lake Zor, which included Eichler's Cove in Newtown, Jackson Cove in Oxford, and of course, Kettletown State Park, By August, we were getting over 5 parts per billion. And in fact, when we did dilutions and we actually did more careful measurements, we got up as high as 22 parts per billion. Now remember, 4 parts per billion is the limit during this time. And so you can bet that Eichler's Cove and Jackson Cove and Kettletown had to close their beaches during those weeks, right, during most of the rest of the summer, because the concentration of microcystin was over that allowed by the EPA and also by the Connecticut DEEP. All these other places, nobody else exceeded, I'm not sure if anybody exceeded over 1, but everybody else was pretty low. Now, that was summer 2017. Summer 2018 was our banner year. Great year for cyanobacteria investigators, because Lake Zor was going crazy high. And so you can see that we are measuring in some places, we're sampling the green stuff on the surface, we are getting over 200 parts per billion of microcystin. Remember, the cutoff is still 4 parts per billion. You got to get people out of the water. So now we're up to greater than 200. And that might seem high to you guys, but you know what? Out in California and in places like Ohio, the Lake Erie, they're up to I've seen numbers up to the hundreds of thousands parts per billion. Tens of thousands to hundreds of thousands of parts per billion of microcystin. So they have it, crazy high out there. And I can't even imagine what it'd be like to be living around places like that. But you can see that, every year from 2016, 2017, 2018, it just kept spiraling up higher and higher. Now you think that 2019 would have been, oh my gosh, are we going to reach the cyanobacterial apocalypse at this point? 2019 was a big dud. I mean, it was great for the recreational users, but for those of us who study it, we're like, I can't tell you how the faces of my student researchers, my student research team, they're all like coming in saying, oh, nothing exciting is happening. I said, well, if you think human deaths and pet deaths are exciting, I guess that's true. But, at least it's low. Everything was low. And Beesick Lake got over 5 in the end of August. But remember, by this point, the threshold is now up to 8 parts per billion, right? So you got to reach 8 parts per billion before you can legally tell recreational users to get out of the water and stop using it. Summer 2020, last year, well, Brookfield's pretty low. Actually, Canobood Lake as a whole tends to be fairly low. I mean, we haven't seen anything big. I mean, you see some blooms of cyanobacteria, but nothing really toxic. Although Lake Zor again, we're almost pushing it. You got up as high as 6.8 in Jackson Cove. And the health director there said, oh, we're just below the limit, where we could have shut down the beach. I said, yeah, well, you can make a call, but legally, we haven't exceeded the threshold yet, right? But in the old threshold of 4 parts per billion, we would have shut down Eichler's Cove and Jackson Cove, right, for sure. So what can we do about cyanobacteria? Well, there's two ways of looking at this, right? If you're looking at, you can look at the fact that we have cyanobacteria here. So if we want to get rid of them, we got to figure out a way of killing them, right? And then the other thing we have to consider is, how do we prevent future outbreaks of cyanobacteria? So we really have to think about 2 things, right? One is controlling existing toxic populations. In other words, how do we kill them? And how do we kill them safely? Because there are ways of killing them non-safely. In other words, we can literally bomb a body of water with poisons that kill everything in there. And that'll get rid of the existing cyanobacteria for a while, but it'll also kill everything else in its place, right? All the other aquatic life will also be wiped out. So we want to think about how to kill them safely. And then, of course, the other thing is looking at future toxic populations. How do we prevent them, right? And to prevent them, we really have to think about, what do they need to grow, right? What can we do to stop them from growing into these giant masses of toxic cyanobacteria. So these are two sides of a similar question here. So let's talk about how to control them. Can we just kill them with things like algicides? And one popular algicide is copper sulfate. You may have heard of copper sulfate before. It's commonly used to kill all sorts of bacteria, sometimes used to kill various types of invasive species in bodies of water. One other type of treatment is percarbonate, which is basically a form of hydrogen peroxide, right, a strong oxidizing agent. And so this one lab decided they would test all three of these chemical algicides to see if they could reduce the concentration of toxin produced by these microcystin populations. What they found was kind of interesting. What they found was that, yes, it did wipe out the number of cyanobacterial cells, right? So that sounds like it's really positive. But the cyanotoxin levels remain constant. And you wonder, like, wait, if we're wiping out the cells, why are the toxin levels still constant? Well, it turns out that most of these cyanobacteria actually store toxin inside of them. And so if you kill the cells, you actually release more toxin into the water. Now, of course, if you happen to be like a dog and you're swimming in the pond and you get all the cyanobacteria stuck on your fur and you're busy licking it off, you're ingesting not just the water, but you're ingesting the cells themselves, right? So you're getting the whole batch of toxin inside of you. You're getting the toxin outside the cell as well as the toxin inside the cell. And so that's really, really bad. So what we're trying to get at is that if we try to just kill the cells, you may not actually be reducing toxin in the short run, right? Short term. You're merely just blowing them up so that now we have more toxin in the water, and that's not a good thing. So we really have to think about controlling them, maybe by prevention. And there are a couple of things that cyanobacteria really love. First of all, they love heat, right? They love heat. So as our summers get warmer and longer due to climate change, we're going to continue to see more and more of these blooms of cyanobacteria. In fact, that's what the whole country has been seeing over the past decade. They've just been seeing increasing amounts of cyanobacterial blooms every year as the summers get longer and hotter. The other thing that cyanobacteria love are food, as we all do, and fertilizers contain lots of good food for them. So we think about agriculture, and we all like to blame agriculture. They're putting all that nitrogen fertilizer out there and it washes them. Well, don't forget our lawns, right? Our lawns are a big deal. And we all put down our turf builder in the spring and then maybe in the summer again, because, you know, Scott says, you got to put it in the spring, summer, and fall, right? 3, maybe 4 applications, if I remember correctly. And so we put a lot of fertilizer on our lawns to keep them looking pretty. But you know what? A lot of that washes off into our waterways, which of course feed the cyanobacteria. Another thing that besides nitrogen fertilizers, phosphates are the other preferred food of cyanobacteria. They love phosphates. Now, some of you may know that if you're planting grass seed for the first time, the starter fertilizer usually contains phosphate because phosphate helps roots to develop. So lots of new lawns going up, lots of phosphorus and phosphates going into the water, into our soil, gets washed into the waterways. TSP, for those of you who have ever done painting, you use TSP as a cleaning agent for cleaning down your walls or whatever it is before you paint. Trisodium phosphate, that's what the P stands for. And then of course, some detergents, not very many these days, but there are some detergents that still contain phosphate because phosphate's a useful cleaning agent, hence TSP. Right, so these are sources in consumer products that contain phosphates that can, again, feed cyanobacteria. And these are things that we can control, right? Whether or not we want to allow these things to go into our waterways. Then, of course, plain old sewage, right? Good old poop. So think about how many people have septic systems, right? Lots of septic systems around in our area. Whoops, let me back up. Oh, sorry. Lots of septic systems in our area. And if you don't maintain your septic system, your septic field well, and things break down properly. Every time it rains, we flush all this stuff into the waterways. And in fact, fecal bacteria, right? The health departments always test for fecal bacteria in our waterways after heavy rains because they expect that sewage is going to get washed in there. Well, it's not just the fecal bacteria, of course, that we have to worry about, but the fact that all that sewage also serves as a food source for cyanobacteria. So again, there are a couple of things we can do to control the future growth of cyanobacteria. There's even climate change, right? Climate change is tied into this as well. And we have to seriously think about how we are responding to climate change. I don't care what people say about, oh, I'm going to deny that climate change is happening, even though half our country is burning up, the other half are, suffering all kinds of weird climate situations. This is happening, and this is affecting cyanobacteria as well. So these are things that are human controlled. There are certain things we can control as humans that could mitigate some of this growth of the cyanobacteria, but it's going to require really, people being committed to taking care of some of these things. So we have to worry about whether or not this is going to be our future. Yes, I know that was a shark, but that could just as well be the scene from, our recreational users when cyanobacteria are floating up to the surface, and we're really, really concerned about our kids being poisoned by the toxins there. So these are pictures of my students that I've worked with over the past 6 years, really. A great group of people who have really been excited to realize that the research that they're doing is not just, generating interesting facts of science and knowledge and stuff like that, but it's actually benefiting their communities by helping protect us from these toxic cyanobacteria. So I really appreciate all the work that they've done. I want to thank all these different, you always see these acknowledgments, right? All these different people, as well as all these different organizations and towns and their health departments and so on for supporting this kind of work. So we will break now for Q&A.
Okay, thank you so much for the presentation. I do have a number of questions that have come in already. And for anyone who joined a little late, we're going to do the Q&A through the chat function. So if you have any questions, please feel free to type them in there and I will ask them of Dr. Wong on your behalf. Okay, so the first question is, what causes these blooms? Why do they occur? So,
right. So again, it goes back to the thing about having all the right environmental parameters that favor their growth, whether it's of course warm summer heat. They love heat. So as the water warms up, they're going to grow. Cold weather they don't like cold weather. Nutrients, again, in the water, just like all of us, right? We all will grow if we're fed, and they will also grow if they're fed. Now, one of the tricky things is, again, as hopefully I made clear in my talk, was that just because we see them growing and they're growing in very dense numbers, doesn't necessarily mean that they're producing toxin, right? And that's always been one of the trickier things that research has been trying to figure out, like, what's the trigger? There are probably several triggers that turn on the production of these toxins in these cyanobacteria. So they've studied, the gene control mechanisms and all this kind of stuff. And it seems like there's nutrients certainly play a role in that. Cell density might also play a role in that. There could be some other factors as well that we're still trying to decipher that actually tell these cells, okay, now we can start making this toxin. Now, I want to point out one thing. These toxins, cyanobacteria do not produce these toxins as a weapon. it's never been found that, there's a purpose for these toxins. These are actually simply metabolic byproducts. Just like for us, a metabolic byproduct is carbon dioxide that we breathe out of our lungs after we've broken down sugar. And for them, some of these toxins are actually just metabolic byproducts of their biochemistry, and they just produce the stuff and dump it off and so on. And so it's just part of their normal biochemistry, and it just happens that these things are dangerous to us, to animals, and to humans.
There were a couple of other related questions, and I'll get to them, but someone just put in a comment that I thought it would be interesting to share, which is that Brookfield experienced a bloom off North Lakeshore Road as late as December 4th last year. So, I mean, just the heat question and the fact it happened in December. So.
Yeah, right. So, yes. So some of these late, like that thing in October, remember back in October 2014, that caught my interest. And I thought, I didn't know cyanobacteria were even a problem in Candlewood Lake, and all of a sudden it's popping up in October when the water is really cooling down. So again, cyanobacteria they're native inhabitants of these bodies of water and our soil and things like that. And if it's a warm December, I can imagine that some of them might still be chugging away. Yeah.
There were a couple of questions related to this. One is the amount of sunlight a contributing factor to increase the amount of toxins in the water? So I guess, it must increase the temperature. So is that, and then do some of the, some lakes that have more tree coverage that lower the temperature, do they have lower levels?
Yeah, so when we do sampling, we also monitor the temperature of the water. And definitely water temperature increases the number of cyanobacterial cells. That seems to track pretty well. Toxin levels, though, do not track with temperature. So again, it's the same thing I mentioned before, that you can have lots and lots of cells, but either very few of them have the genes for producing toxin, or if they do have the genes, they're not turning on those genes to make toxin. So that the temperature doesn't track with the toxin levels, but it does track with cell number. And of course, if you have a lot of cells, and if they just happen to have the genes for it and they all turn on, that's going to be a lot worse than if you have very few cells and they have the genes and they turn on, right? So large cell numbers is still should be a concern, right? Because you don't want to have a lot of these guys around, because just on the off chance that they happen to have toxic capabilities, now you're going to produce a whopping amount of toxin in the water.
People are curious about the testing, and do municipalities test regularly? Can members of the public access sort of weekly or regular measurements of toxin levels? And then on a related note, why doesn't Connecticut test for anatoxin in recreational waters? You mentioned that they did not. So what would be the reason for that? So first, are there regular measurements? Where can people find them? Are they, are they measurements that people can actually use to make decisions? And then why isn't the state testing for anatoxins?
So for the first question, the Candlewood Lake Authority gets our numbers, and if you look on their, I believe it's their Facebook page, which means you need Facebook. You might be able to access it through their website, but I'm not quite sure. I've always seen it on their Facebook page. But Candlewood Lake Authority will publish the weekly toxin reports for Candlewood Lake towns, which include Brookfield, on their Facebook page. The Lake Zor Association, Friends of Lake Zor or Lake Zor Association, also publish the Lake Zor results, which are Jackson Cove, Eichler's Cove, and Kettletown, on their website. I'm not sure about what goes on with some of the other lakes, like Beesick. I mean, their lake association gets the results, and I hope that they're letting people know about that. But at least for Candlewood, and Lake Zor, they do make it accessible to the public to get a hold of. Why aren't we testing for anatoxin or saxitoxin, which is even more, right? There are more cells or more places have saxitoxin genes than anatoxin genes. And saxitoxin is just as lethal as anatoxin is. Well, because again, as you saw, it's kind of rare. It's rarer, not that rare, but it's rarer than microcystin is as a toxin. And so it just doesn't get tested. And why don't they? Well, I've talked to the people at DEP and they often tell me, we don't have the money. I say, okay, I understand that. You got to get funding to do these kinds of things. And yeah, so if we want our waters tested for other kinds of cyanotoxins, especially ones where we see in the news that it's killing pets and even people, maybe we should be testing for those things. But again, it's one of those things where, well, it's not that frequent. It's kind of rare. So we won't include it in our budget item because we just can't afford it at this point, or it's not a priority. Unless people really want to, say, hey, we need testing for this type of thing. If somebody wants to pay me, I could probably test, but it's really expensive to test. The reagents, the chemicals, and the equipment needed for this it's not cheap. It might be cheaper to test for COVID than it is to test for cyanotoxins.
Someone asked if one area of a lake has high levels, does that affect the rest of the lake? I know, can it be localized within a body of water?
Yeah. So when you're talking about a lake like Candlewood, which is huge, it's 8 miles long, sometimes we'll find high levels up at one end, like up in Sherman. I remember Sherman and New Milford a couple of weeks ago had rather not super high levels, but they had certainly had blooms up there and higher levels. And then you go down to New Fairfield and Brookfield, there was nothing. It was really clear. And so when you have huge bodies of water like this, certain places will get they'll have different quality of water, right? You've got runoff maybe going on up in the northern end of the lake that's feeding the cyanobacteria that's not taking place down in the middle of the lake in the southern end of the lake, or vice versa. So in huge bodies like that, you're not going to have what's affecting one part of the lake way up there may have no effect, down in other parts of the lake. Now, if you have a smaller body of water, like a pond, it can be more homogeneous and more uniform. You'll probably see, it's one part generates stuff here, you'll see something in another part. The Housatonic River, which is Lake Zor, right there, it's a river, right? So you've got water, even though it's a dam down there at Stevenson Dam, water is still flowing down the river. So you've got input coming in all the time. You've got outflow, and that'll also affect whether or not toxins build up in a certain area or cyanobacteria build up in a certain area. They actually don't like rapidly flowing water. They prefer to just be in still water, which is why ponds and lakes are really places that often get it, or little coves right within rivers. That's why Kettletown State Park's beach, I don't know if anybody's ever been to Kettletown State Park Beach, but it's in a really deep cove, and there probably isn't a lot of water circulation in there, which is why it gets really, really juicy in the summer. I wouldn't want to go in there.
I do want to say that I received a comment that Brookfield funds testing at the Town Beach Weekly, yes, and reports high levels on the health department website.
Okay.
So just it's important to note. So let me ask, are there toxins found in the fish that are caught in the bodies of water that you've been testing? So if someone catches a fish that's been in an area with high.
Yeah, so there have been some studies about how much what an effect having cyanotoxins in the water would have upon game fish that are caught and so on. Um, there really, I've not seen like any major. There doesn't seem to be like a concentration of toxin, in the flesh of game fish that are in these waters. Like you would expect from mercury and things like that, where it accumulates. Um, but nevertheless, right? I wouldn't want to eat a fish that was swimming around in this dense green cyanobacteria laden body of water. I mean, I'd worry about even being out there and fishing in that area again because of the one of the really interesting areas of study now is in aerosolized cyanotoxin. University of New Hampshire is really big on that. They've got a huge program where they're studying aerosols of cyanotoxin, and they find that cyanotoxins get blown up into the atmosphere, quite a distance, in fact, up into the atmosphere. So you think about, you're out fishing, somebody comes by on a boat, right, churning up the water, throwing up some spray. You don't think much about it, but what they've done is actually thrown up millions and millions of cyanobacterial cells up in the atmosphere, and even wind and so on, right? They'll throw up these. Remember, these are microscopic cells, so they get thrown up into the atmosphere. You're breathing it in. Again, this is where it ties into the thing about the Lou Gehrig's disease and people living near bodies of water they're not even touching the water, but they're just living within like a mile or two of the body of water that has lots of cyanobacteria. And now we see large numbers of neurodegenerative diseases. We have to be concerned about breathing in this stuff. So even if you're not eating the fish, I think just being out in the water when there's large populations of cyanobacteria, that could be of a concern in itself.
The question I wanted to ask, and you sort of touched on it here, is about that connection, the potential for a connection between cyanobacteria and neurodegenerative disease. So you mentioned the University of New Hampshire is looking at that. But I'm wondering, what types of studies are being conducted on this? I mean, it's if there is a causal connection, it's quite important to know about, right? So who's looking at it? Are there any preliminary data? Are there ongoing long-term studies, or
right?
What's happening with that?
Yeah, so this is still sort of a correlation study, right? There, when it comes to these things, they're saying, oh, look, these people live near areas that have large numbers of cyanobacteria. We can detect cyanobacteria in the air, and there's a much higher rate of neurodegenerative disease among these residents, right? So that's not obviously a cause and effect right there. It's correlation study. But from animal studies, they do know that, okay, if you take this neurotoxin, you administer to this animal, whether it's through gastric introduction or whether it's intraperitoneal into the skin or into the muscle, we see the liver fail when it comes to microcystin, or we see the neurons die, within certain doses of this neurotoxin. So those are just, right now, of course, we can't extrapolate these. We can't definitively say that these are going to happen to humans. We extrapolate them from animal studies. And then, of course, the folks down in Brazil, those poor people who are just having their kidney dialysis, I mean, they died horrible deaths from liver failure. And that was an introduction of the microcystin toxins literally right into their bloodstreams in the kidney dialysis. I mean, so if you don't want to, if you want a giant study, there it is, right? There's a huge number of people but that's, again, that's from introducing it into their bloodstreams through the kidney dialysis. That's not the same as, oh, I'm playing out in the water on a nice day at the beach with my family, and I'm getting some of this on my skin, and maybe a little bit splashing on my mouth. How much of it is dangerous? Maybe it is not an acute danger. Maybe it's a chronic danger. Maybe it's a long-term thing. Okay, I wiped out some liver cells. I wiped out some brain cells from that day at the beach. And it's just going to build up over time every time I go there.
There are a couple of more questions, and I don't want to keep you too late, so I hope you'll entertain a couple more. um, One is someone wanted to know, are cyanobacteria or the toxins, I guess, but are cyanobacteria present in bodies of water that are used for public water supply? And if so, can the toxins be removed successfully?
Yes, yes. So they can get into reservoirs because I mean any place that it's an environmental body of water, right? So soil cyanobacteria, aquatic cyanobacteria, they'll eventually get in there. Can it be removed in waters from the water supply? So I mean, I've seen reports where water treatment systems claim that they can remove this stuff. How effective that is, not quite clear. Um, Again, Lake Erie, I'm sorry, Toledo, Ohio, back in 2014, their water treatment system was overwhelmed by the amount of cyanotoxin that was coming through it. The kidney dialysis unit in Brazil, they depended on their water treatment system in that city to clean out the toxins, and it didn't work. Right? It wasn't effective. So all these people died as a result of it. So yeah, reservoirs can also get contaminated with cyanobacteria. And so water treatment facilities have to be very, very conscious of those types of things. They should be monitoring the water and so on. But I remember reading some study where they weren't, they were looking at some water treatment, some facilities and some reservoirs, and they weren't testing for cyanotoxins, or they weren't monitoring it at all. They weren't even monitoring whether there was any presence of cyanobacteria. So, I can imagine in some places fecal bacteria is our main thing. I got to worry about poop washing out of sewage and into the water. We're not thinking about cyanobacteria, which can be even worse or just as bad.
Actually, two more questions and a comment. That's okay. So someone asked, is there an evolutionary benefit of these specific bacteria? So what is the...
Yeah, they make oxygen.
Right.
It's been predicted. I remember once I saw a number that said maybe up to like 25% of our atmosphere is, 25% of the oxygen in our atmosphere is produced by cyanobacteria. I don't know if that's how close that is to reality, but they still play a role in doing photosynthesis. And a lot of this is actually some of the marine cyanobacteria, but more so than the freshwater ones because there's more marine area. But yeah, they do that. And of course, those that fix nitrogen, they're actually contributing nitrogen compounds which are beneficial to plants and other types of things that grow in the soil. Yeah.
I just don't know. I'm going to show my lack of knowledge, but is it unusual for bacteria to create oxygen? I mean, usually you think of plants creating oxygen. Are there other types of bacteria that create oxygen, or is this unique?
You have to be photosynthetic to do that.
And so, there are photosynthetic bacteria. There are many types of them. Or?
Well, so that's why the cyanobacteria are the primary photosynthetic bacteria. Okay. In fact, it's believed that, so we all know that plants have these little organelles, these little compartments in them called chloroplasts, which are the main factories for capturing sunlight, generating oxygen, and then producing sugar along with that. Right? That's how plants work. And what's a lot of interesting studies have looked at chloroplasts and found that they have all the same features as cyanobacteria. And so it's believed, in fact, they even have DNA that's like the same size as that found in cyanobacteria. So it's believed that chloroplasts, in fact, used to be some kind of cyanobacterium that invaded, a plant, an early plant cell, and kind of settled into a nice symbiotic relationship and said, I'll make food for you, I'll make sugar for you if you give me a safe place to live. And voila, there we have photosynthetic plants arising from photosynthetic bacteria.
Very interesting.
Endosymbiotic theory.
So the another question and then a comment, like I said, sorry, I had to ask about I just didn't know about the oxygen and bacteria. Okay, so someone writes that she sees cosmetics that list blue-green algae, maybe perhaps similar to what you're saying about the supplements, and she's wondering if, wondering if it's toxic to the skin or can using them affect your health in any way. And then we can open that up wider to have there been any incidents with the supplements that that show some sort of health problem for your health based on use of them?
Yeah, I guess I haven't really been paying much attention to the supplements that much. I mean, except for that one study that I was looking at that measured, rather significant levels of microcystin within some of these health food, blue-green algae health food supplements. I think the thing is, whenever you're dealing with supplements, like I said before, right, it's not regulated by the Food and Drug Administration. So pretty much Joe down the street who has a farm pond can say, hey, I've got some blue-green algae growing in there. I'm going to scoop it up, dry it out, package it, and sell it as something. They can do that. So even if it's hopefully the company that's making this, the cosmetic company that's making this, is actually testing these things on a regular basis, right? You have no idea if they are or not, but you hope that they do. I mean, it'd be nice if they admitted that, yes, this is part of our regular safety protocol, that we do test these ingredients coming in and making sure that they're not toxic, etc. But there's no regulation for it. So anybody and their mother could be selling supplements, and you, as a consumer, consumer beware, right? Buyer beware. So be aware that, yeah, they're selling this stuff and we know the potential for danger. So
could it be if it's used in cosmetics and it goes on the skin, it could affect you, the toxicity could affect you. If the toxicity were present, it could affect your skin or.
Yeah. So some of these toxins are also, you might have seen in the list that I showed you, but some of them are dermatoxins, which means that they cause skin rashes. In fact, a lot of people think that, when some people go to the beach, and they come in and say, I got this rash on me, from being out on the water that day. Some of that is probably due to some of these dermatoxins, these skin toxins that are produced by cyanobacteria. And hopefully you're not eating it or anything like that. Or if you breathe it, what if there's saxitoxin or anatoxin in there and you're breathing in some of that cosmetic, on a regular basis? Is that going to be slowly degrading your brain cells? I don't know. It depends on whether there's actual toxins in the product, whether the producer is actually testing these things for safety or not. Hopefully they do.
And then the last, if anyone else has any questions, now would be the time to type it in. But the last thing is a comment, and it's actually from the health director for Brookfield, who's on the call. And he had a couple of comments. I didn't want to identify him, but I do want to identify him for this one. And that's that he writes, please know that if you see blue-green algae at the town beach, it has not as yet meant that it is toxic. So you don't assume just because you see it that it's toxic, blue-green or cyanobacteria.
Right. Yeah, that's the point I've been trying to get across, right? We can't tell just by visual inspection of the water that it's dangerous. In fact, recently there was a homeowner, I think in Brookfield, who saw a bloom, sent in a sample that was tested. It looked pretty, it was fairly juicy. It wasn't, very, very heavy, but nevertheless, you could definitely see the cyanobacterial cells in the water. And, the toxin levels were really low. So again, you can't just do it by visual inspection, right? Because they might have the gene, they might not have the gene. If they have the gene, they may not be turning it on. All these different things, right, that come into play. So don't panic. But panic, I don't know. It's hard to say panic, but don't panic. Be cautious, but don't panic, I guess.
That's a good rule. And I want to thank you very much for sharing your research with us tonight. And I hope that you'll come back and share additional research as you have it, or so I think it was really helpful for residents to hear from you tonight. So thank you very much.
I want to thank you all for putting up with me for this whole time and looking at data. I mean, sometimes raw data, which is scary at times. But thank you for your interest, and I mean, enjoy our waterways. Just be a smart recreational user, whatever that means. Don't make sure you wash off. Make sure you wash yourself after you come out of the water. Don't lick your hands.
Okay, well, thank you so much. And as I said, we recorded tonight's presentation, and it'll be available on the Brookfield Library YouTube channel in case anybody wants to watch back or share it with someone you know. And okay, well, thank you so much.
Thank you. Thanks,
Good night,
everyone.
good night, everyone.
Have a good evening.